Good morning, students. Myself Ramesh, Botany NEET faculty. Today we are discussing about the important topic in 12th biology. The topic name is molecular basis of inheritance. Right? This is a very important topic in 12th biology. That's a molecular basis of inheritance. Right? This is lesson important for the NEET by because the 4 or 5 questions coming from this topic. Now every year very important topic means this is the base of the some lessons also. That is the reason this is a very important topic we are calling. Right? Here see in the molecular base of inheritance. Right? This word already we listened in the before lesson that inheritance and variation we discussed there. Right? Now what is the meaning of the inheritance? The inheritance means the characters are that carry forward to one generation to the another generation. The similar characters carry. Right? See here. We can write the similar characters carry to one generation to another generation. Right? What is the meaning of the one generation to another generation? That's we call the parents to progeny. Right? Means the parents to children. They transfer the characters, the similar characters. That's we call the inheritance. We call it. Right? We can write down the parents to progeny. Right? Here see what is the reason for the inheritance. Right, we discussed the genetic material transferred to the one generation to the another generation. Means the genetic material carry the characters one generation to the another generations. Means what is the reason for it? Means the inheritance they carried through the genetic material. What we call it, this is the genetic material. In this lesson, we are discussing about the what is the genetic material, what is the structure of the genetic material, and what is the functions of the genetic material. Right? Basically, the nucleic acids we are considered as a genetic material. Nucleic acids we are calling they are the genetic material. Right? See here, the how many nucleic acids are there? Mainly the 2 types of nucleic acids are there. Right? We can write the nucleic acids. 2 types are there. One we can write the DNA. Another one is the RNA. Right? Both are we call the nucleic acids, and both are act like a genetic material. Right? Now we see the what is the DNA. Already you know the deoxyribonucleic acid. This is the DNA, and the RNA means we call the ribonucleic acid. Right? DNA means deoxyribonucleic acid, RNA means ribonucleic acids. Right? See, here is the DNA. We call the means DNA is the main genetic material for the all living organisms. We call it. Right? The DNA, the most of the living organisms, DNA is genetic material. Not all. We are not mentioning the all. The most of the living organisms, DNA is the genetic material. Right? We have written the most of the living organisms. Most of the living organisms, DNA is genetic material. Most of them means the almost all the like the plants and animals, the humans and the microbes, like the DNA is genetic material. But not all. Most of the living organisms, DNA is the genetic material. Right? We can see the RNA. Here is the RNA also genetic material, but some virus only, not all living organisms. Right? Not all viruses also. The RNA is the genetic material, only some viruses only. Here is this RNA act like a genetic material, some viruses. Right? We can take for example the TMV virus. Right? This is the one plant virus. In the TMV virus, the RNA is a genetic material. Right? In the TMV, this is the plant virus. Means we can say the tobacco mosaic virus. In this one, this RNA is the genetic material. See here, the RNA they are the act like a genetic material some viruses, but they are perform the some other functions also. RNA they are act like only in viruses as a genetic material, but they have the some other functions. Like what are the functions we can see? They can act like a messenger. They act like a messenger, means they transfer the genetic information like one generation to the another generation. That's we call the messenger. And they act like a adapter. Adapter means in the protein synthesis process, they can pick up the amino acid to transfer to the protein chain, right? That's become adapter we call it. This also they have function. And also they have the catalytic property also. They means they act like an enzyme also. That's we call the ribozyme enzyme here. The RNA also we call the ribozyme enzyme, means they digest the RNA. Right? These are the functions of the RNA. We call them the RNA is not main genetic material, means it's genetic material only in some viruses. They also they perform the other functions. That's we call the messenger, adapter, and catalytic property. The catalytic property we can take the ribosome. The ribosome digests the RNA. Right? This deoxyribonucleic acid and most of the living organisms, DNA is the genetic material. Now we can see what is the history of the genetic material, how they identify the genetic material. Right? Now we clearly the genetic materials are two types. One is the DNA and RNA. Right? The both are we call the genetic material. Both are we call the nucleic acids. They will ask, for example, question: which is the genetic material present in the most of the living organisms? You go with the DNA there. And which is the genetic material in some viruses only? That is the we have to go the RNA. The which genetic material they have the catalytic property? You have to go with the RNA. Like this, you have the questions they will ask like this. The questions are from this topic. Now we have to use the history of the DNA, the who identified and the who given the name of the genetic material. Right? See, first of all, the scientist we can take the Friedrich Michel. In Friedrich Michel scientist in 1869, the scientist name you have to write the Friedrich Michel in 1869. This is also very important. 1869 in pus cells. Pus cells means we call the leukocytes. In the pus cells, in the pus cell nucleus, inside the nucleus, he identified the genetic material. He identified the genetic material, and he don't know the name. He not given that like nucleic acid. Just he was simply he called the nuclei. Right? He called it that. He called it that is the nuclei. Right? The first the genetic material identified by the scientist name we can take the Friedrich Michel. The year is the 1869. And which cells he identified? He identified in the first cells. In the first cells, the inside the nucleus, he identified the genetic material, and also he given the name of the nuclei. And after that, in 1889, the scientist name we call him the Altman. The Altman, they given the name of the nucleic acid. He called that is the nucleic acid. Why? Because they have the acidic property. Because of acidic property, he called that the nucleic acids. Right? Altman giving the name of the nucleic acid. What is the reason for this one? Is that they have the acidic property. Right? They identify the Friedrich Michel, but he don't know the name of the nucleic acid. They simply called nuclei. Why? Because he is present in the nucleus. Right? After that genetic material have the acidic property. The Altman given the name of the nucleic acid. And one more scientist there. The scientist name we call the Khorana. Khorana means Hargobind Khorana. In 1954, in this year, the early years. 1954, he synthesized the artificial DNA in vitro condition. Like in test tube, he synthesizes the artificial DNA. The corona we call it. Here is the, he synthesized the artificial DNA. Right? Here is the, that's also we call in the artificial chromosome also. In which condition that's we call in the, in test tube, he synthesized this we call the vitro condition. And for this one, in 1957, he got the Nobel Prize also. He got the Nobel Prize in the 1957. These are the scientists in NCERT given, like one is the Friedrich Miescher in 1869, the first cells. Then 1889, the scientist name we call the Altman, and he given up the nucleic acid name. And after Corona, in 1954, he synthesized the artificial DNA. Right, see, already we know about the based on the nucleus development. The nucleus fully developed, which is the early cell. There is mainly two types cells are there. One we call the prokaryotic cell and eukaryotic cells. The two types cells are there. Now we are discussing about the what is the structure of the prokaryotic cell and what is the structure of the means. We have to mainly discuss in the what is the genetic material in these two cells. The prokaryotic cells, which type genetic material is present, what is the structure of that one, and eukaryotic cells, and which type genetic material is present, what is the structure of that one. Right? Now we can see the prokaryotic cell genetic material. The prokaryotic cells means already you know what is the prokaryotic cell. Undeveloped nucleus. We call it the undeveloped nucleus presence. Means the nucleus membrane is absent. That type cells we call the prokaryotic cells. We call it. See here the prokaryotic cell. The prokaryotic cell. The prokaryotic cell means in this one we have to say the nucleus membrane is absent. That type cells we call the prokaryotic cells. Nucleus membrane absent. Means the genetic material naked presence in the cytoplasm. Right? We call the naked genetic material. Naked genetic material in cytoplasm. And in this DNA, they have the circular DNA is present. This is the cat prokaryotic cells. Means the double stranded circular DNA is present in this one. Double strand circular DNA. Right? We can say the DNA of the prokaryotic cells like this. They have the circular DNA and like these structures present. Right? The double strand circular DNA presence in the prokaryotic cells. And also the specially in some prokaryotic cells, the extra chromosome circular DNA is present in some prokaryotic cells, not all. We mentioned some prokaryotic cells. Here is some prokaryotic cells. What they have means extra chromosomal circular DNA is present. Circular DNA. This extra chromosome circular DNA, what we call that is calling the, we calling that is the plasmid. This extra chromosome circular DNA we call the plasmid. Right? They have the like this like structures present in the means some prokaryotic cells one present, some two present, like the more number of the plasmids also present there. Right? This we call the extra chromosome circular DNA we call it. This how the self replication capacity also present in this one plasmid. And these are used in the recombinant DNA technology process, used as a vectors. These plasmids used as a vectors. Means in these lines we discuss the prokaryotic cells. The DNA is the circular DNA is present. Circular DNA is present. And here is a double stranded circular DNA. And some cells have the extra chromosomal circular DNA is present. That one we call the plasmid we call it. right? The plasmids used as a vectors we call it. This is about the prokaryote cells, right? Now we can see that what is the eukaryote cells DNA is present, right? In the eukaryote cells, we can see in the eukaryotic cell, what is the mean of the eukaryotic cell? Well developed nucleus present, well developed nucleus, that cells we call the eukaryote cells, right? The prokaryote cells example we can take the bacteria, the monoras, we call the prokaryote cells. The eukaryote cells, the plants and animals, right? These are we call the eukaryotic cells, we call the plant cell and animal cell, that cells we call the eukaryote cells. Eukaryote cells means the well developed nucleus is present, means nucleus membrane is present, that genetic material presence inside the nucleus, not in the cytoplasm. This is the difference. We can see that here is the nucleus developed means well developed. We can write a well developed nucleus present, right? In this one, the genetic material presence inside the nucleus. Here is a genetic material present inside the nucleus, not in the cytoplasm, right? In the prokaryote cells, that presence in the cytoplasm, but eukaryote cells, they presence inside the nucleus. In the eukaryote cells, they have the like a linear DNA is present. The DNA not circular, right? They have the like the linear DNA is present, like this structure they have, right? This we call the linear DNA. Double stranded linear DNA is present in the eukaryote cells. And some more things are there in some cell organelles, like you can take the mitochondria and chloroplast, right? Here see some cell organelles, that is a mitochondria and chloroplast. Already you know the what is the function of the mitochondria. Mitochondria perform the respiration process, that's we call the aerobic respiration process, and the chloroplast that perform the photosynthesis process. These two cell organelles, they have the like the circular DNA is present in this one also. In this one now the circular DNA is present. Circular DNA is present. These two cell organelles we call the semi-autonomous cell organelles we call it. Means they are replicated, but they are depending on the nucleus for the replication process, right? That is the reason these two cell organelles we call the semi-autonomous cell organelles. Here autonomous means totally they are freely, they are not depending on others, that's we call the autonomous we call it. Semi-autonomous means they have the freedom, but they need some guidance and some energy from the other cells, that we call the semi-autonomous cell organelles. This chloroplast and mitochondria both are, they can perform the replication process, but they are depending on the nucleus. That is the reason these two are we call the semi-autonomous cell organelles we call it, right? In this one, the circular DNA is present. These are the DNA types that we can take. The means the prokaryote cells, they have the circular DNA is present. Eukaryote cells, linear DNA is present. The prokaryote cells, they have the extra chromosomal circular DNA. What we call that is that's we call the plasmid. The plasmids using as a vectors in the recombinant DNA technology process. And the eukaryote cells we call the linear DNA is present, and some cell organelles like chloroplast and mitochondria. In this both they have the like the circular DNA is present. Because of this circular DNA, right? The cell organelles actually the cell organelles totally the DNA is absent. The ribosomes also they not the genetic material is absent, like that we have the like. But especially in this mitochondria chloroplast, they are the like semi autonomous cell organelles we call it. Now we can see the length of the DNA. What is the length of the DNA? The how the DNA's length we can measure, right? Means the length of DNA based on the number of nucleotides, number of base pairs. The how many nucleotides present? 1 present, 2 present, 3 present. Means based on the more number of present means the more length the DNA is present. Right? So you can see the length of DNA we can see now. Right? Here is the length of DNA. Right? This length of DNA based on the number of nucleotides. Based on the number of nucleotides. Number of nucleotides and based on the number of base pairs. Base pairs we can using the symbol of the BP. BP means base pairs, right? One base pair is equal to two nucleotides. We consider. Here is a one base pair is equal to, we can write the two nucleotides, or also we consider the two nitrogen bases. Two nucleotides and two nitrogen bases are there. One base pair is equal to two nucleotides, two nitrogen bases. Right? The how many nucleotides present, how many nitrogen bases present, the how many base pairs present. That's we consider the length of the DNA. Right? For example, some scientists given like some numbers are given. These are also very important. Which one they have, like bacteriophage, the Psi 174 given. The numbers are given. We can write the four examples given here. What is the number of nitrogen bases? What is the number of base pairs present? Right? Here, for example, we can take the bacteriophage. Bacteriophage Psi 174. What is the meaning of the bacteriophage? We can write is the strain type. The bacteriophage means which virus attack the bacteria. That virus we call the bacteriophage. We call it. What is the meaning of the phytophages? Which virus attack the plants? That's we call the phytophages. And zoophages also we call it. Which virus attack the animals? That we call the zoophages. Cyanophages also we call it. Which virus attack the green algae, blue green algae? That we call the cyanophages. Like we have the like bacteriophage numbers given. Which virus attack the bacteria? That virus we call the bacteriophage. The strain number the Psi 174. In this one they have the 5382. 5382 nucleotides present. Means nucleotides numbers equal to nitrogen bases. Like the 538 nucleotides are 5382 nitrogen bases we can write. And the bacteriophage lambda. Bacteriophage lambda. These have the 48502 base pairs. 48,502 base pairs present, not nucleotides. This is the base pairs are present. 48502. And E. coli, Escherichia coli bacteria. E. coli bacteria, Escherichia coli bacteria. In this one they have the like the 4.6 into 10 to 4 of 6 base pairs. It's a very important. 4.6 into 10 to 4 of 6 base pairs present in the E. coli bacteria. And for example, we can take the human genome. If they mentioned genome means the haploid number of DNA content, haploid number, right? Means we cannot consider the 40 chromosomes. This genome means we have the like haploid number, 23 chromosomes only. The human genome that they give the 3.3 into 10 to 4 of 9 base pairs. Means one gamete they have the genome means one gamete have the like the 3.3 into 10 to 4 of 9 base pairs. We can write the total human DNA. Total human DNA they have like 6.6 into 10 to 4 of 9 base pairs. 6.6 into 10 to 4 of 9 base pairs. This is the total human DNA base pairs. This is a very important. Sometimes they ask like simply what is the base pairs in the E. coli bacteria. That's the 4.6 into 10 to the power of 6. In the human genome, they asked human genome applied content. you go with the 3.3 into 10 to 4 9 base pairs. If they ask total human DNA means you go with the 6.6 into 10 to 4 9 base pairs. Right? This is about the, you have to write the, what is the number of nitrogen bases. What is the number of base pairs? For example, I am asking in the E. coli, they have the 4.6 into 46 base pairs present. Here see, the bacteria lambda, bacteriophage lambda, the how many nitrogen bases are present? For example, I am asking, how many nitrogen bases in bacteriophage lambda? Here how many we can write simply? Here base pairs are present. One base pair is equal to 2 nitrogen bases. You have to simply write the 48502 into 2. They are the nitrogen bases. That is the given clarity. Right. The next we have seen the how the DNA is formed. How the DNA. What are the components present in the DNA? Right. The DNA we call the polynucleotide chain. We call it. DNA is the polynucleotide chains. Means group of the nucleotides they can form the DNA structure is formed. Right. Now we can see the what is the polynucleotide chain, how the DNA is formed. This is the introduction of the molecular base of DNA. Right. This is the introduction part. The DNA they have the like different type of metabolism they can perform. Like we can take the replication process, the transcription, translation. There is a DNA perform the functions. This is enough. The next class we discuss the what is the structure of DNA, components of the DNA. Correct.